So remember that DNA replication is any time you want to make a new copy of DNA. So we're going from DNA, making another strand of DNA. So if I have A, C, T, the other side is going to be T, G, A, right? So I can keep my T's as long as I'm going from DNA to DNA. I can keep my A's and T's because thymine is in DNA. So when I look at this, for example, this is the DNA strand I had. I'm going to make this bigger. So this is the DNA strand I started with. It was unzipped, and then DNA polymerase, this big beige glob structure, is an enzyme that is bringing new nitrogen bases and dropping them by base pairing down one side of each strand. So I end up with two new strands at the end. One has a version of the old strand, right? Has this old strand that was retained, and then it has a new strand that was replicated. So what do we call this process when each new strand has an old strand and a new strand? We say that it is semi-conservative, right? Semi-conservative. So whenever I need to make more DNA, basically making another cell by mitosis, and mitosis happens when we're growing from a zygote, when sperm and egg first come together and that cell divides and divides and divides and divides until you have a fetus ready to be born, there's a lot of DNA replication going on during that time. When we shed our skin cells, there's a lot of DNA replication going on during that time to make new skin cells. What were you going to comment?
Does that ever screw up?
Oh, yes. It does, yeah. DNA polymerase does make mutations, but it's good at fixing them as well. When a cell has a mutation, that particular cell doesn't have what it needs to reproduce and survive. But sometimes they do, and then we have tumors. That's when we think of what cancer is, and that's going to be a future topic coming up. It's just DNA replication gone wild. So you have DNA replication and then cell division. And when those cells are dividing out of control with mutations, that's cancer. So that's another example of where we would see DNA replication. So anytime we're replacing cells, making new cells, repairing cells, DNA replication has to occur so those new cells have a copy of the DNA from the pre-existing cell. Yes? Yes, yes. Helicase unwinds the DNA molecule. DNA polymerase drops new nitrogen bases to make the new DNA strand, and ligase connects nucleotides on the lagging strand of the DNA molecule. So ligase, to ligate means to tie in medicine. If you ligate something, you're tying them together. So just think as ligase as putting things together after DNA replication is done, or at least as it's moving along down the strand. Ligase ties it together, and then helicase unwinds to get it started, and then DNA polymerase is the boss of actually dropping those nitrogen bases down. Yeah, good question. So the unwinding, helicase, right? Complementary base pairing is DNA polymerase, and the joining of those on the lagging strand, where we have those fragmented pieces, that's ligase. So that's, we already highlighted that, that's that. So replication then is making new DNA. And someone asked, does it make mistakes? Yes, about every 100,000 base pairs. But there's always there's checkpoints. When cells get ready to divide, they undergo checkpoints to make sure there's no major errors and that those cells are healthy and ready to divide. And we can correct those errors, which is pretty cool. So remember, DNA exists to make proteins, to make new proteins. So now we're going to talk about the next process, and that is transcription. So transcription is not making more DNA to replace cells. Transcription and translation are processes for making proteins. So when we need to make a new protein, we go through the process of transcription and translation using the information from DNA. So transcription is when we're taking DNA and making messenger RNA from it. So we're not going to use T's when we're making messenger RNA, right? What do we use instead? What? U. Yeah, if we have an A on DNA, we're going to drop a U when we're making RNA. So that's what this strand here is messenger RNA. And remember, every 3 nitrogen bases on messenger RNA we call a codon. And every 3 nitrogen bases on DNA we call a triplet. It's not labeled here, but I think it's on a future slide. But it's called a triplet. This is called a codon. And then we take this messenger RNA. Where is this occurring? Where does transcription occur? Make sure you highlight that if it's not already. Yeah, transcription occurs in the nucleus. And translation occurs at the ribosome. So translation is taking messenger RNA and dropping amino acids. Remember all these here at the bottom are amino acids. So the goal of translation is to take messenger RNA and make amino acids, or drop amino acids. And how do the amino acids get there? What other RNA plays a role here? Yep, transfer RNA. So transcription is going DNA to messenger RNA. Translation is going messenger RNA to tRNA to amino acids. And then you link those amino acids. What's the name of the bond that links these amino acids together? Peptide bonds. Those are special covalent bonds that hold amino acids together in a protein. Yes?
So is a triplet the same as
A triplet, yes, except for what are you going to see? Because anticodon is tRNA, right? No, you're right what you said. The nitrogen bases are the same, except for transfer RNA is going to have uracil and DNA is going to have thymine, right? Yeah. Okay, so again, whenever we're making uracil or RNA, we're going to put uracil in there. There's 3 types of RNA. Messenger RNA, again, where is that formed? In the nucleus. And ribosomal RNA is out at the ribosome. And transfer RNA is hooked up to an amino acid out in the cytoplasm. All right, so transcribing, again, is going from DNA to messenger RNA, and translating is going from messenger RNA to tRNA to amino acids. So we looked at transcription. Every 3 DNA bases is called a triplet. Every 3 messenger RNA bases is called a codon. So this process here is transcription because I'm making a single-stranded messenger RNA right here. You can see it being formed. The purple strand is the single-stranded messenger RNA. And this is the unwound DNA molecule. And I can see I'm making messenger RNA because here's an A on the DNA and I dropped a U instead of a T to make my messenger RNA strand. Then this messenger RNA strand is going to be complete once we hit that stop codon, and it's going to break free, and a couple more things happen after that that removes the exons and keeps the introns, which is the nonsense DNA is spliced out, but that's beyond the scope of what we need to know. So then that messenger RNA comes out, hooks up with the ribosome, and this large transfer RNA has an anticodon sequence. These 3 are specific to this amino acid, and that hooks up to the messenger RNA molecule by base pairing. This is called the anticodon, right, because it's on transfer RNA, and this is called the codon because it's on messenger RNA. So this here, again, this pink structure is our messenger RNA, and this is all happening at what organelle out in the cytoplasm? Out at the ribosome, yep. Okay, so remember the ribosomes has those 3 sites where that transfer RNA comes in. There's the A site where it comes in, and then it slides over, and the peptide bond forms between the amino acids, and then it exits. That empty transfer RNA exits and goes off and finds another amino acid floating in the cytoplasm. So there's those 3 sites. It arrives here, we can see, and then the peptide bond forms to this growing chain of amino acids at the P site, and then it exits. So we have this chart of amino acids. There's 20 amino acids in this chart. And these all come into our bodies through our diets, right? We bring in different proteins. Our digestion breaks them down into different amino acids. And we have all these building blocks to reassemble to make different proteins. So we labeled this diagram in that worksheet. Does everybody still have that worksheet? Make sure you have that ready on Thursday so you can turn that in. Or at least use it as a review before you take the test. I don't have it with me. I can give it to you after class. There's one next door, I think. So be able to label what's going on here and understand what's what. Because here's my nuclear membrane. Here's the cytoplasm out here. And there's animations in our folders, too, that puts this all in motion and talks about each step of the way. So if you're not familiar with this, just take the time to visit it again. OK, so with that, I want to play a little Kahoot review game. And I'll put this in our Blackboard so you have that as a review as well.